Okay, I am going to try to use this diagram to illustrate the Bohr and Haldane effect and explain the ionized bicarbonate reaction to you guys. I think trying to understand that from listening to a recording and looking at a PowerPoint is challenging, so I'm hoping that this diagram will help to simplify it. I also did a diagram just kind of demonstrating the transport of oxygen and then the transport of CO2. So oxygen is transported two ways as a refresher. One way, it's dissolved in plasma, which is the very small, kind of inconsequential way that it's transported. The major way that oxygen is transported is attached to a hemoglobin. And then for carbon dioxide, it's transported three ways. The first way is dissolved in the plasma, which is about 8%, a little bit more than oxygen, but still a pretty small percentage of all of its transport. The second way that CO2 is transported is directly bound to a protein like hemoglobin, and that's about 12%. And then the major way that CO2 is transported, about 80% of its transport, is through this ionized bicarbonate reaction. And I think it seems more complex than it really is, and I'm hoping that this diagram will simplify it a little bit for you guys. So that's what we're going to talk about here. So I just want to draw your attention to a couple of things. Over here I have the lungs, and I'm emphasizing this high pressure of oxygen in the lungs. And the reason why I want to emphasize that it's a high level of oxygen here is because this is where we're going to be loading the oxygen. We're going to be loading it onto the hemoglobin, and we're going to make oxyhemoglobin. All right. This is also, if you were thinking back to the oxygen dissociation diagram, this is also a left shift in the oxyhemoglobin dissociation curve. All right. So this is where the affinity of a hemoglobin for oxygen is increased. Decreased affinity or increased affinity for oxygen. It wants to load oxygen, and that's due to that positive cooperativity, right? The more oxygen it has, the more oxygen it wants. So in the lungs, your hemoglobin very much likes oxygen and it binds oxygen readily. The other thing that helps that, the other thing that makes oxygen want to load in your lungs is that you have a high pH or an alkalotic pH. High pHs are alkalotic. You have low CO2, low H plus ions, and then you also have this low 2,3-DPG, which we're not really going to emphasize, but just knowing that it's a low 2,3-DPG for that. So all of these conditions make this a loading zone for oxygen, right? And that oxygen's going to bind with the hemoglobin to make this oxyhemoglobin, or HbO2. And then you can follow this over here, HbO2. When it gets to the tissues, you now have this low PO2, right? This is not a place where there's a lot of oxygen. Your tissues use up the oxygen. That's how they function. So this is an area of low PO2. This would be a right shift on the oxyhemoglobin dissociation curve. And a right shift means you have a decreased affinity for oxygen, which means no longer does this hemoglobin like the oxygen. It doesn't like it. It wants to unload this oxygen. It's going to unload that oxygen into the tissue cells, right? And the other reason that the hemoglobin's affinity for oxygen decreases is because in the tissues you have a low pH or acidotic. So low pH is acidotic environment, low pH, a very high CO2, a very high H+, and then you have high 2,3-DPG, which you don't really need to know again, that just memorize that it's high. These are more the components that I want to draw your attention to. So all of these make it an environment where that hemoglobin no longer likes oxygen. It wants to unload that oxygen. And then what it does do is it makes it an ideal environment to load CO2, which is really what we want to be taking out of the tissues back to our lungs. So this ionized bicarbonate equation, the main way that CO2 is transported goes down like this. So you have CO2 leaving your tissues, and it's going to go into your red blood cells. And when it does that, it's going to combine with water. The CO2 and the water then make something called carbonic acid, which is that H2CO3. And this is that compound that does not want to stay together, okay? This wants to dissociate, which means it wants to break apart. And because we're in this condition where oxygen is not readily available, we have this acidic pH, CO2 is readily available, H plus is readily available, we're going to dissociate in a very specific way. We are going to dissociate and form HCO3, or this is known as bicarbonate, and that's going to leave the cell. Chloride is going to come back in, and that's just a balancing of charges, and this is that hamburger phenomenon, and I'm not going to talk any more about that except for just that exchange is called that. And then it's going to also form H plus ions. Now these H plus ions are really kind of what I want to draw your attention to, because they bind with the hemoglobin to make HB. So now you have this hemoglobin that is binding to H plus. And if you remember back to the fact that we talked about how hemoglobin are essentially buses that can carry all kinds of gases. They can carry carbon dioxide, they can carry oxygen, they can carry this H plus ion, they can carry all kinds of things. The only thing that's important to realize is they have to unload something to load something. So here they unloaded the oxygen, they loaded the CO2, this ionized bicarbonate reaction happened, and now you've got this HHB going back to the lungs. So now we're in the lungs, right? So imagine that we've got this going back to the lungs, but now our environment has changed. Now we have an alkalotic environment with lots of oxygen, a high pH, low CO2, low H+. And what happens is that pressure of oxygen is high enough that it comes down here and it kicks that H+ ion off, right? So it kicks it off the hemoglobin. So now you have this free-floating H+ ion that's no longer attached to the hemoglobin because the oxygen kicked it off and made HbO2 again. So now that you have this H+ ion free floating, it attracts that bicarb back into the red blood cell. So this HCO3 minus comes back into the red blood cell. It joins to that H+, and it forms that weak acid again that we know doesn't like to stay together. It forms that H2CO3. That does not want to stay together, right? We know this. But now because we're in the lungs and the environment is different, we have this high O2, high pH, all these things are different. Now it's going to go into CO2, back into CO2 again, and water. And this, my friends, is what we exhale. Okay, so that CO2 and that water goes into our lungs and gets exhaled out. So that is how the ionized bicarbonate reaction works. So I feel like it looks really complicated, but it's not too bad. So you have this CO2 plus H2O plus water that goes to H2CO3, and this is that weak acid that does not want to stay together, and then that can go to H plus bicarb. So in the tissues, because you have low oxygen, the affinity for your hemoglobin is low for oxygen. You have this acidic pH. This is going to be the Bohr effect. This is going to be the way that equation dissociates in the tissues. And this is going to form that HHB and affect your transport that way. When you come over to the lungs, this is your Haldane effect. And that same reaction is going to happen, but now the other way, and you're going to get that CO2 and H2O to exhale out the lungs. And the big difference here is hemoglobin's affinity for oxygen and what's available. So in the lungs, lots of oxygen available. Hemoglobin's affinity for oxygen is higher. Not very much CO2 available, so hemoglobin isn't really trying to bind CO2. Over here in the tissues, low oxygen, not a lot of oxygen available, so hemoglobin is not going to have a great affinity for oxygen. It's not going to want to bind it. Lots of CO2 available, so you're going to see this HHB or this dissociation go this way. So that is how this works, and it goes both ways depending on where you're located. And I hope that this really cleared it up. I know it's a tricky concept, so we'll definitely talk about this in class, but I hope this diagram kind of helps to explain it.